I'm live. I'm back, and I'm here to talk about a whole bunch of stuff. I might might break into a lot of coughing fits because I was sick. I got sick again. So I got sick over Labor Day weekend, and it was pretty bad. But it was just more like flu-like symptoms. Like I got super tired, and then my whole body started like hurting. And then I guess sore throat, and then like runny nose, right? And then I got better, but then this week I picked back up with body aches, like intense body aches. Past 2 nights I couldn't sleep at all because I kept like waking up, and it just hurt so bad. And now I'm just like I'm completely better, not tired, not anything. It's just I can't stop coughing, and it's like a really dry cough. I literally went through 3 packs of these. This is my fourth one. I went through 1 pack today. Like each day I go through a pack. So like I already took out like 3 of these. So I went through a pack and 3 cough drops. And these are the big ones too, which said a lot, and they're not working because I'm still coughing. I'm like trying to drink water and have cough drops, but I'm still coughing. So if I break into a coughing fit, don't don't do that. Please don't do that. Anyways, I just figure out that like a bunch of people know that I'm like streaming now, which is it's okay. I'm not like exactly complaining, but what it means is I have haters now. Like, oh hey, don't hate on me for my bed, bro. Like if you think if you're gonna make comments about my bed being messy, just quit. Just don't watch my lives anymore. Also, I got some haters talking about how I should change my shirt. I like this shirt. Don't talk about that. Like I like this shirt. I'm gonna wear whatever I want, and I make these lives for me. So don't don't bring don't bring my room and my shirt to this. Bring the material. If you have anything to comment about the material, if I got anything wrong, let me know. But my fashion and my decor, whether I keep up with it or not, is none of your business. Okay, got it? Okay. And no cheesecake this time. It's just me and my water. If I get hungry, I have pimento cheese in the fridge, so I might get it out and then eat it on my next live. I plan on doing 3 lives today. First one is gonna be this worksheet, and then I'm gonna do anthro, and then I'm gonna do the next lecture on bio because I have an exam this Thursday, and I want to do good on it. Hopefully, okay. Okay, so getting into this, I already. Oh, by the way, I'm gonna label this like quiz 3 or whatever. I already took the quiz, but I'm just gonna review because I need to review. I don't know anything about this. All right, so this lecture is going to be all about protists, and of course, this is one of the domains in the eukaryote kingdom, and there's 4 protistance supergroups. There's excavata, yeah, then SAR, so like SARS, and it does stand for something, but we're gonna get into it later. And then there's Archaeplastida, and then there's Uniconta, okay? And the basically what the word supergroup means is it's a large group of organisms with common ancestors and unique characteristics. So there's 4 like supergroups that group protists into. Another thing, I have like a relationship term for you. You ready? Since a lot of people don't have healthy ones, here's a relationship for you to digest. It's that's not yours. So this is endosymbiosis. The word endo means in, and we already learned about symbiosis. So this is a relationship where one organism lives in another, and I'm dying. Um, maybe I shouldn't talk while I'm sick, but guess what? my test is Thursday, so I'll have to talk. I'll try to make this quick. One organism within another, so it could be like parasitic, or mutualistic, or just commensalistic, commensalistic, commensalic. Okay, whatever. Just get over it. Okay, so first we're gonna get into the supergroup Excavata. There's 3 phylums. There's the Diplomonads, and then there's the Parabasalia, and Euglenozoa. Okay, so let's see. Okay, so the Excavata, all of the phylums, they have flagella, so they're like classified by flagella, and they have like modified mitochondria. Sorry. So the first phylum, the Diplomonads, they have mitosomes, and what mitosomes are they just are modified mitochondria for the Diplomonads. They have 2 equal-sized nuclei, so they're kind of like they have 2 binucleic. I don't know if that's a word. Anyways, they have multiple flagella, of course. Example is Giardia lamblia, and it causes giardiasis, which is an infection, causes nausea and diarrhea, and it's common in poor sanitization and unsafe water. So yeah, very nice. Don't come across that. All right, so the next phylum is Parabasalia, and it's a parabasal organ. It has a parabasal organ, which is similar to like the Golgi, and then there's a hydrogenosome, which is in the mitochondria, I think. But anyways, it's there's hydrogen produced as a byproduct of making energy for these organisms. There's multiple flagella. Example organism is Trichomonas vaginalis, and it causes trichomoniasis, which is an STD, which we love, right? And or trick, and the individuals often are asymptomatic at first. I feel like I'm doing something wrong here. I'm gonna have to like review because I'm not sure. Anyways, the next phylum of the Excavata supergroup, there's Euglenozoa. So we got 2 species we're talking about. There's Pyranema species, which is colorless and clear. There's no chloroplast, but after they eat, they can be green. There's no chloroplast though. Just keep in mind. There's 2 flagella, of course, and the green color due to food vacuole contents comes from algae. I'm dying. And okay, so we're gonna move on to the Euglena species. The species of Euglena. Okay, when I say species, I mean like it's the group, and then there's species after. Anyways, so it's the most studied group in the Euglenozoa phylum, and it's found in fresh and salt water, both fresh and salt water. And most, but not all, have chloroplast, I believe. And they have a stigma, which is a red pigment spot that expands when it reaches certain light wavelengths. So basically, stigma are activated from stimuli. So it's red. There's 2 flagella. I'm about to cough again. And it moves from phototaxis, which is, of course, the stigma responds to light, and so it allows it to it allows the Euglena species to move. Positive phototaxis is moving toward the stimulus, and negative is moving away from. And there's a 9+2 microtubule arrangement. So basically, it's kind of like a circle. Like basically, the microtubules, like there's a pattern. So there's there's 9 microtubules in like the periphery of the circle, and then there's there's 2 in the middle. So it kind of looks like a face, like a circle, and then two like dots in the middle, basically, because of the microtubules, and it allows this microtubule arrangement allows for tip to base undulation, which is kind of like the way an inchworm moves, basically. The last thing to talk about the phylum Euglenozoa, but we're going to talk about the kinetoplastids. So they have a modified mitochondria called the kinetoplast. They have at least one flagella, and an example organism is the Trypanosoma brucei, I think, and it causes African sleeping sickness in vertebrates bloodstream. And the symptoms include lethargy and malaise, and this carried carried by the tsetse fly, spelled TSE. How do I cough? How do I stop coughing? Let's search up how I stop coughing. Oh, I hate this. Apparently, I'm supposed to try a humidifier, but where am I gonna get a humidifier? Please enlighten me. It's okay. Anyways, we're done with the supergroup Excavata. We'll move on to the SAR supergroup. So, what does about to forget? Straminophiles and alveolates and rhizarians. So that's what SAR stands for. So straminophiles, alveolates, and rhizarian. Yeah. Okay, so straminophiles. What does it kind of sound like? It sounds like straw, right? So they're kind of known like straw hair organisms. So the flagella described as fine and hair-like. But these include diatoms and brown algae and oomycetes. DBO. Or BOD. BOD. Nice BOD, bro. If I'm talking very slow today, it's because I'm trying not to cough. I feel like restrict my. Anyways, so the diatoms are photosynthetic and unicellular, and they're made of silicon dioxide. The diatoms, and they actually help produce diatomaceous earth. And then the brown algae are multicellular and mostly marine. Many are just simply called seaweed. So yeah, brown algae is like seaweed. So the components of seaweed, there's the holdfast, the stipe, and the blade. So holdfast is what anchors it down to the ground. Pretty simple. Name speaks for itself. The stipe is the stem, and the blade is like leaves. Like it's like not actually leaves. It's like a leaf-like structure. And the brown algae actually goes through sporic meiosis. And they're heteromorphic, which means sporophytes and gametophytes are structurally different, and isomorphic would mean they're structurally the same. Please don't cough. Please don't cough. Okay, we're gonna move on to the oomycetes now. So it quite literally means like egg fungus. So this group, well, tiny group, used to be classified as fungi because they have like hyphae which are like branching filaments. You want to know what hyphae looks like? Kind of looks like neural system. Anyways, so an example organism of that would be Phytophthora infestans. Yeah, but it's actually it turns tomato and potato plants into black slime, which is pretty nice. And it was responsible for the Irish potato famine, which is also nice. But yeah, thing to know about. Actually, let me just finish all this and then I'll um, go over. Oh, we're gonna move on to ovules anyways. Okay, so basically, diatoms are made of silicon dioxide, and they have they make diatomaceous earth, and they look pretty cool. Brown algae, basically seaweed. They have the hold fast the site in the blade, go through spore meiosis, and yeah, they're heteromorphic. And then the oomycetes, they used to be classified as fungi because they have hyphae, but their cell walls are actually made of cellulose, not chitin, so they're not fungi. Oh, excuse me. So we have alveolates, and basically all of these have alveoli, which are membrane-bound sacs. We have three types. Three types of alveolates. We have dinoflagellates, apicomplexa, and ciliates. So the dinoflagellates, we already kind of went through this in like the lab. I already talked about it in the lab, but we're gonna talk about it now because why not? Okay, so we have an example organism of a dinoflagellate, which is we have Noctiluca, which is sea sparkle. Pretty nice. It's in the ocean and it has a bioluminescence. And the example organism that causes toxic blooms, we have Gymnodinium. This one's a little less fun and happy than sea sparkle. It depletes the water of oxygen, so like the fish die and stuff. It's pretty nice death. But guess what? Dinoflagellates also have longitudinal and circumferential flagella, which means they also have undulation. Undulation, sorry. The next group is Apicomplexa. So they have an apical complex to aid in host cell penetration. So these are parasites. Apicomplexa. Parasites. We don't like those. And an example organism is Plasmodium. And what does Plasmodium do? It causes malaria. Pretty nice. But something about malaria you should know is the primary host is actually the mosquito, and that secondary host would be us, humans. The third group of alveolates is ciliates. They have ciliary beating from cilia, and they have cilia for movement and feeding. But anyways, the ciliary beating is gradual, controlled movement. And there's two examples. I didn't write down the other one, but I have paramecium. So yeah, just know that, I guess. But there's three parts of this, I believe, of the ciliates. There's the cytosome, the cytopharynx, and the cytoprop. So the cytostome is the mouth, the cytopharynx is the throat, and the cytoprop is the anus. And they have also osmoregulation, which is contractile vacuoles function. All right, we're going to move on to the R of SAR, the rhizarians. So many of these are amoebas that have fake feet called pseudopodia. We have three groups too. We got radiolarans, which are usually marine, and the shells are made of silica and often symmetrical. They have cytoplasmic streaming. Whoa, like me. But I don't stream cytoplasmically. I stream like YouTube. But it allows for carrying captured prey into the main part of the cell. So I think, yeah, I don't know. We're gonna move on. Forams. So they're like foraminiferans. and it means like little hole. They have porous shells called tests, but these are made of calcium carbonate. They're strong, and 90% of all identified forms are known from fossils. And then we have cercozoans, which are they have thread-like pseudopodia, and they're found in freshwater, marine, or soil ecosystem. And we have Polynella species. It has a chromatophore, which contains peptidoglycan and allows for photosynthesis. So we just went through the Sauria group and we went through the Excavata group. Something to know about straminophiles, they have flagella, and they like find here like flagella. So we have diatoms, silicon dioxide. Wait, I already talked about that. Okay, so we already talked about straminophiles. So straminophiles again with the find in here like flagella, diatoms, brown algae, and oomycetes. And then the alveolates, we have the dinoflagellates, the Noctiluca and the Gymnodinium. They're kind of like polar opposites. We got Apicomplexa, which are parasites with malaria and stuff. Ciliates, which have cilia, obviously. And Rhizarians with the pseudopodia. We got the Radiolarians, forms, and Cercozoans. But yeah, they all have pseudopodia. Okay, and then we have the fourth supergroup. It's the Archaeplastida supergroup. This contains red and green algae. So the brown algae belongs to the stars. It belongs to the S, which are the straminophiles, which have the straw cilia. No, flagella. Okay, so we've got red algae, which are also known as rhodophytes. They have phycoerythrin, which is a red photosynthetic pigment, and it masks the chlorophyll green. And examples of these would be the nori, carrageenan. Please don't cough. We got green algae, which are Archaeplastida. Wait, I already said that. Anyways, we got chlorophytes. They come in many forms. We got sea lettuce, which is ova, and then we have charophytes, which are the closest relative to living plants. So chlorophytes and charophytes. Okay, last supergroup, we got Uniconta. So we have amoebozoans and opisthecons. Amoebozoans have a lobe or tube shaped pseudopodia, and we got we have tubulinids, slime molds, and entamoebas. Okay, so the tubulinids, an example organism would be Amoeba proteus, and the slime mold we have Plasmodium versus cellular. So Plasmodium is bright yellow orange. It forms a mass called Plasmodium. Again, not to be confused with Plasmodium that causes malaria. But it's bright yellow orange, forms a mass called Plasmodium. It's in soil, mulch, and rotting things. So cellular slime molds are individual cells, and they're just individual. They're not like, they don't form a mass or anything, but they're clear. They look like slugs. Anyways, the slime molds are mycetozoans, which is kind of like, it means like fungus animal. So they produce fruiting bodies and they produce fruiting bodies which aid in spore dispersal. So yeah. Since I think slime molds can move, that's why it's like called fungus animal. But fungus because they produce fruiting bodies and fungi produce fruiting bodies. Okay, we have the entamoebas, which is symbiotic parasites. So the entamoeba histolytica causes amoebic dysentery via water or food. So yeah, if you want to get dysentery, get you some entamoeba histolytica. Last group of the supergroup Uniconta, we got the Epistoconts, and they contain more protists, fungi, and animals. Just to keep it simple. But yeah, that was just a worksheet. I will actually write this all on my board because I no way I'm gonna remember all this. But yeah, I'm glad to have reviewed, and I will end this right here and then move on straight to Anthro.